Cell theory is the cornerstone of all biology, providing the fundamental framework for how we understand life itself. Modern cell theory is built upon two simple but powerful truths. First, that every living organism is composed of cells, and second, that all cells arise from pre existing ones. By recognizing the cell as the fundamental unit of life, science transformed our ability to diagnose disease, understand evolution, and explore the mechanisms of existence. In 1838, the biological sciences were transformed. Botanist Matthias Schleiden, observing plant tissues under a microscope, made a profound discovery. Shortly after, in 1839, physiologist Theodor Schwann explored animal tissues. He realized that animals, just like plants, were composed of cells. Together, their work established the foundational principle of biology. Cells are the fundamental building blocks of all living things. Before 1852, the scientific community was confused about where cells actually came from. Many scientists falsely believed that cells formed spontaneously, crystallizing out of organic fluids like salt in water. In 1852, Robert Remak revolutionized biology. He provided definitive evidence that cells do not just appear, instead they divide. He proved that cells originate only from other pre-existing cells, a core principle known as biogenesis. This concept, later popularized as omnis cellula e cellula, became the foundation for modern cell theory. In 1855, Rudolf Virchow popularized the phrase cellula a cellula, or all cells come from pre-existing cells. This idea became a pillar of modern biology. While Virchow gained immense fame for spreading this discovery, he failed to credit Robert Remak, who had provided the initial definitive evidence 3 years earlier. This historical oversight sparked a long-standing debate regarding scientific integrity. It remains a poignant reminder in research today. Progress depends not just on discovery, but on recognizing the work of those who paved the way. Why do mitochondria and chloroplasts act so differently from other cell parts? They contain their own DNA and divide on their own. The endosymbiotic theory explains that these were once independent, free-living bacteria. Eventually, they entered a symbiotic relationship, being engulfed by an ancestral host cell to become permanent residents. This union transformed these organelles into the powerhouses and energy factories we recognize today. Konstantin Mereshkowski set the stage in 1905. He hypothesized that the chloroplasts in plant cells were not original parts of the cell, but ancient photosynthetic bacteria that had entered into a symbiotic relationship. In the 1920s, Ivan Wallin took the theory further through experimentation. He attempted to culture mitochondria in isolation. While his work was met with skepticism at the time, he provided the vital experimental groundwork for future investigation. Decades later, in the 1960s, the scientific community finally had the tools to prove it. Advanced genetic sequencing revealed that the DNA inside mitochondria and chloroplasts is unmistakably bacterial in origin, finally cementing the endosymbiotic theory as fact. By the mid-1960s, the idea that complex cells evolve through symbiosis was widely considered an obscure, unproven hypothesis. In 1967, biologist Lynn Margulis revolutionized our understanding of life. She synthesized vast amounts of data, combining evidence from microscopy, fossil records, and early genetic analysis. By weaving these disparate threads of evidence together, Margulis turned a neglected concept into a foundational pillar of evolutionary biology. Her work proved that endosymbiosis was not just a curiosity, but a major driving force of evolution, forever changing how we view the origin of complex cells. Before the 19th century, medical experts believed in the miasma theory. They were convinced that diseases were caused by bad air or rotting organic matter. This belief persisted for centuries, leading to ineffective public health measures. However, through observation and rigorous experimentation, scientists eventually dismantled this idea. They discovered that invisible microorganisms, not bad air, were the true culprits behind disease. This paradigm shift, known as germ theory, forever changed medicine, public health, and our understanding of life itself. In 1546, long before the discovery of microbes, Girolamo Fracastoro had a revolutionary intuition. He proposed that diseases were caused by seed-like particles, which he called seminaria. He suggested these tiny agents could spread between people through direct contact or even through the air. Though he was centuries ahead of his time, his brilliant ideas remained dormant, waiting for future science to catch up. In 1847, physician Ignaz Semmelweis worked in a Vienna hospital where he observed a terrifying trend. He noticed that doctors frequently went straight from performing autopsies to examining expecting mothers, without any cleaning in between. Semmelweis hypothesized that cadaverous particles were being carried on the doctor's hands. He insisted on a rigorous hand-washing protocol with chlorinated lime. The results were immediate and drastic. Death rates plummeted. Yet, despite this evidence, his peers rejected his ideas, unable to accept that they were the cause of the infections. In 1848, London faced a terrifying cholera outbreak. At the time, the world believed diseases were caused by miasma, or bad air. A physician named John Snow took a different approach. He mapped every death in the neighborhood, noticing a chilling pattern. The cases were clustered around a single public water pump on Broad Street. By removing the pump handle, he stopped the spread of the disease. This was the birth of epidemiology, proving disease spreads through contaminated water, not bad air. Snow's map became a foundational tool for public health. In the 19th century, many believed in spontaneous generation, that life emerged from non-living matter. Louis Pasteur designed a brilliant experiment to prove otherwise. He created flasks with long curved necks. He boiled the broth inside to kill any existing microorganisms. The curved neck allowed air in, but trapped dust and microbes in the bend, preventing them from reaching the broth. As long as the flask remained undisturbed, the broth stayed clear and free of life. This showed that life only comes from pre-existing life. When Pasteur broke the neck, allowing microbes to enter, the broth rapidly spoiled, confirming his theory and laying the foundation for modern germ theory. In the mid-19th century, operating rooms were dangerous places. Surgeons often moved between patients without cleaning their tools, unwittingly spreading deadly infections. Joseph Lister, inspired by Louis Pasteur's work, hypothesized that these infections weren't caused by bad air, but by microscopic germs. Lister introduced carbolic acid to disinfect surgical instruments and wounds. This simple shift toward antisepsis fundamentally changed the survival rates of patients. By proving that microscopic germs were the enemy, Lister saved countless lives and launched the era of modern sanitary surgery. Today, we apply these theories in every doctor's visit. From staining a slide to using a stethoscope, our ability to diagnose illness relies entirely on the foundational work of these pioneers. These tools represent the final evolution of the science we have been exploring. Historically, we guessed that diseases were caused by vague forces like bad air, known as miasma. Today, germ theory allows us to use precision tools like pulse oximeters to measure oxygen and cultures to pinpoint specific pathogens. We have moved beyond guessing. By identifying the exact pathogen causing pneumonia, healthcare providers can transition from vague theories to targeted life-saving treatments. Cell theory, endosymbiotic theory, and germ theory might seem distinct, but they are bound by a common evolution in how we study life. The link between them is a shift toward rigorous observation and replication. This scientific standard moved us from guessing to truly understanding biology. Today, this synthesis is the foundation of modern healthcare. It allows doctors to use diagnostics to identify specific pathogens and provide precise, life-saving treatments. Science is a deeply human endeavor, often marked by both brilliant discoveries and historical controversies. Two such figures in 19th century cell theory are Robert Remak and Rudolf Virchow. Remak provided the first definitive evidence that cells derive from other cells. However, Virchow later popularized this concept, essentially claiming the fame while failing to credit Remak's earlier work. This incident serves as a vital reminder that scientific progress relies on more than data. Integrity, rigorous peer review, and proper attribution are the essential pillars that keep the pursuit of knowledge honest and reliable. Science is a collective generational effort. Ensuring ethical practice is not just a formality, it is the very foundation of our progress. While these theories have stood the test of time, the frontier of biology continues to expand. Today, genomic research is revealing deeper connections between our own cells and the ancient endosymbiotic events that made complex life possible. Advanced genomic mapping is uncovering how our DNA still carries the echoes of these ancient bacterial mergers. Furthermore, we are only just beginning to understand the human microbiome. This vast ecosystem of bacteria within us is not just a collection of stowaways, but a critical partner in our survival. As we peer into the future, we see that the story of life is truly a story of cooperation, reaching back billions of years to the first cells. From Robert Hooke's initial observation of cells to the complex genomic medicine of today, our understanding of life has evolved through centuries of discovery and necessary skepticism. These theories, born from careful observation and rigorous experimental evidence, remain the essential pillars of modern biological study. Together, they provide the framework that allows us to diagnose, treat, and understand the intricate systems that make up a living organism. As we look to the future, these foundational stones continue to support the next era of biological insight.